All right, we can get started now. I'm Rachel Hostetler. I'm a scientist at the Allen Institute for Brain Science, and I'll be moderating this webinar and also doing some intro to the ABC Atlas itself. And then Chris Morrison is a software engineer at the Allen Institute for Brain Science as well, and he'll be going over some of the Jupyter notebooks and how to access data in the ABC Atlas programmatically. And as a reminder, this webinar is being recorded and it will go up on our YouTube channel afterwards. And this webinar is actually part of a webinar series. So if you'd like to join some of our future webinars, please remember to register for them. We also have time to take some questions, so please post them in the Q&A section on Zoom. And if you're watching a recording of this, you can always post your questions online in our community forum. And the link to the community forum is posted in our YouTube description. And we're from the Allen Institute, and the Allen Institute is a bioscience nonprofit research institute in Seattle, Washington. And the main principles of the institute focus on team science, big science, and open science. And so with team science, we have very large teams similar to core facilities who do different parts of the experimental pipeline. And with that, we're able to accomplish really large projects with a lot of robust data that is massive amounts, in the terabytes of data. And we share all of that data openly on our website at brain-map.org because we believe in open science and sharing our protocols, data sets, preprints, anything we can think of online on our website. And today specifically, this is what we'll be talking about. We'll be talking about how to access the single-cell RNA sequencing and some of the MERFISH data that is featured in our Allen Brain Cell Atlas from some of our human and mouse cell type taxonomies. All right, so now I'll briefly go over the ABC Atlas, which is an online browser tool that you can access from anywhere with internet, and what data sets and what types of information are featured in this tool. So as I just mentioned, the ABC or Allen Brain Cell Atlas is an online interactive tool which features large taxonomy data sets. And so the data sets that are currently featured in there are the whole mouse brain, which features both single cell and MERFISH data. And there were two publications that contain these data, Yao et al. 2023 and Zhang et al. 2023. And these were both part of a Nature package. And there's also an entire webinar featuring these data sets on the BICAN YouTube channel. So if you have more questions about the methods, how the clustering was done, any of the analyses, I really recommend checking out these two papers. And you could also watch this webinar as well called Whole Mouse Brain Atlas Publication Package Highlights on the BICAN channel. So that's our first taxonomy that was put into the Allen Brain Cell Atlas. It's the entire mouse brain. Then the second taxonomy featured the whole human brain, which is single nucleus RNA sequencing data. And these data came from a paper, Saletti et al. In Science. And this paper was also part of a very large paper package back in 2023. And there's also a webinar on this specific paper package called Human and Non-Human Primate Brain Atlases and Tools on the BICAN YouTube channel as well. And for those unfamiliar, BICAN stands for Brain Initiative Cell Atlas Network. So these projects were done not just at the Allen Institute, but as part of a larger consortium involving researchers and scientists at institutes all over. The human Alzheimer's disease taxonomy, which specifically features data from the middle temporal gyrus and dorsolateral prefrontal cortex. We have both single nucleus RNA sequencing data and MERFISH data from these experiments. And this was just published in 2024 as Gabito and Travaglini et al. And that was published in Nature Neuroscience. And we just had a webinar on this last February, about a month ago, called the Seattle Alzheimer's Disease Brain Cell Atlas. And that is on the Allen Institute YouTube channel. And again, these three webinars that I've called out, they feature the lead scientist on these projects. So if you have, pretty detailed questions, I would really recommend watching these webinars to see if your questions could be answered there. Since each of these projects involve hundreds of people, you might be able to get really detailed questions answered through here or through our community forum. And then our newest data set that was just added to the ABC Atlas about a month ago is single cell data from our Aging Mouse Brain project. And this was just published in January 2025, Jin et al. This was published in Nature, and we'll actually be having a webinar focused just on this project next month, April 23rd at 9:30 a.m. Pacific time. All right, so starting with the whole mouse brain atlas. For those who have not heard about this yet, it was both single cell and spatial transcriptomics experiments done to create this atlas, and the single cell transcriptomics experiments were done first to create a taxonomy, and over 4 million cells were profiled, and then the information from that taxonomy was used to pick the 500 gene panel for the spatial transcriptomic data, and those were integrated together to make a spatial cell type map. And so this taxonomy has many different levels, many different hierarchical levels. And so in the ABC Atlas, we start with our class level, which features 34 classes of cells. Then there's a subclass with cell types nested underneath that. And then we have supertypes nested within those. And then at the very bottom, we have clusters, and over 5,000 clusters were identified in this whole mouse brain taxonomy. And here's what that looks like. So here are UMAP plots, which are displaying the different levels of the taxonomy. So again, in the atlas, we start with 34 classes. Then we have 338 subclasses, 1,201 supertypes, and then at the very bottom, 5,000 different clusters. And so you can view all these different levels of the taxonomy and different subsections using the Allen Brain Cell Atlas, and I'll show you that in a little bit. Okay, our next taxonomy, and then, yeah, some more detail about that. We have, again, there were 2 different papers that were involved in this study, the Yao et al. 2023 and Zhang et al. And so these are divided into a couple different data sets in the Allen Brain Cell Atlas. So first we have our 10x single cell data here, which over 30,000 genes were profiled, and this information came from over 300 different mice that underwent different microdissections of different brain areas. Then our MERFISH data set from the Allen Institute, which was led by Honghui Zhang, that we currently have 1 male mouse brain up on the atlas. This is the mouse brain ID, and there were 500 genes that were used for these experiments, and this is sectioned in the coronal plane of section. And then we also recently added a second data set featuring the imputed genes from the single cell data here as well. So now we have over 8,000 imputed genes into the MERFISH data set. And again, that's because the MERFISH data and the single cell data are part of the same taxonomy. So if you're looking at a cell type in the single cell data and it says this is supertype XYZ, if you find that supertype in the MERFISH data, that's the same supertype or the same cluster or same class or subclass. So those numbers that I mentioned, the 34 classes, so many subclasses, that pattern is the same whether or not you're looking at the single cell data or the MERFISH data. And that's also true for the MERFISH data from the Zhang et al. Paper, because these 2 MERFISH data sets were both integrated into the single cell data set here. So again, any label that you see for any class or subclass or cell type is consistent and has the same meaning across these 3 different types of data because they were all integrated together. And in our MERFISH data from Shaowei Zhuang's lab, we actually have 4 different brains that are up in the Allen Brain Cell Atlas. Some are coronal, some are sagittal, and they had a bit of a larger gene panel as well. And again, these are part of a Nature package here, published in December 2023, a 10-page Nature package. Okay, so now moving on to the aging mouse brain taxonomy. So this is our newest taxonomy that was just added recently. And I'm having this following the mouse brain, the whole mouse brain, because the whole mouse brain data was also used to help create this aging mouse brain taxonomy. And so not the entire brain was profiled for the aging mouse brain, but a number of large portions here, which you can see, different areas. And so you might recognize that the colors on these plots here are very similar to the plots on the previous panels. And that's because if you're looking at a class, subclass, or supertype in the aging mouse brain taxonomy, they were actually mapped to the whole mouse brain taxonomy that I just showed. And since we didn't have every single brain area profiled in the aging mouse brain, we have a smaller number of classes, subclasses, and supertypes. but these levels were mapped onto that taxonomy. The part that's different here in this taxonomy is the clustering. So in this taxonomy, we had aged mice, and then those were compared to young adult mice from the whole mouse brain study. And they were looking at differences in these cell type clusters between the aging associated clusters and the young adult mouse clusters. And so the clustering here was done a bit differently. And again, we're having a webinar specifically on this paper April 23rd. So if you're interested in this paper, please attend our future webinar where we'll have the lead author, Kelly Jin, describing the research and all of their methods. And here's what's in here again. So the aged mice, there were 44 mice that were used for the single cell data. They were aged to 18 months. And then again, they were compared and integrated with the taxonomy and the transcriptomic data from the 64 young adult mice that were used from the whole mouse brain paper. And over 30,000 genes were profiled. And again, that was just published in Nature in January. So you can also read that as well. Okay, our whole human brain taxonomy. Similar taxonomy set up to the other ones, but we have a couple different names. So in the previous taxonomies, we went class, subclass, supertype, and cluster. This taxonomy has a different naming scheme. So we actually have superclusters at our highest level. Our middle level is called clusters, and then our most granular level are called subclusters. And so this paper was taken, these data come from the paper published in 2023, where over 3 million nuclei were profiled from 105 different anatomical dissections. And these are the results of that taxonomy here. And in the ABC Atlas, our neurons and our non-neuronal cells are divided into two different data sets. And for the actual brains that were used for these experiments, there were 3 whole brains and a 4th brain that contained just the primary motor cortex was used, and over 50,000 genes were profiled. And again, we have our neurons and our non-neurons split into 2 different data sets in the ABC Atlas. And this was part of a much larger paper package in Science that featured 21 papers that was published in October of 2023. And now our Alzheimer's taxonomy. Here we have a couple different levels of our naming as well. We have classes, subclasses, and supertypes. And this came from looking at nuclei in the middle temporal gyrus and the dorsolateral prefrontal cortex. And what's unique about this data set is the data from the Alzheimer's donors from middle temporal gyrus was that information in that transcriptomic data was mapped onto a reference atlas of non-pathological donors from the middle temporal gyrus. So we had a reference taxonomy that the cell types were mapped onto. And then because of that, the leads on the team were able to see which cell types were specific to Alzheimer's disease and did not appear in the original reference data. And we can see that here over in the right, we have this arrow pointing to the CAD cell types, the microglia, specifically over here. And that means that these were cell types that existed only in the Alzheimer's donors and not in the original reference atlas, reference taxonomy from non-pathological donors. And a subset of the donors that were used for that Alzheimer's project, 24 of them were also used for MERFISH experiments. And this was published last year in Nature Neuroscience. So here's the paper associated with that. And I mentioned earlier, we just had an entire webinar on this in February. So you can, that webinar features 4 of the lead authors on the study. So if you have more details about the specifics of how they carried all this out, you can look up that webinar and hear from them how it went. And because we had a number of different parameters that were involved in the Alzheimer's study, we have a couple of different filters as well. So this is a screenshot showing what the filters actually look like in the Allen Brain Cell Atlas. You can divide the MERFISH or single nuclease data and filter it based on all of these different categories here. And so the goal of this project was to have a variety and the entire span of Alzheimer's disease. So there were some people who had high levels of pathology or very low levels, some with very high levels of dementia, some with no dementia at all. So we had about 50% cognitively normal in our cohort and 50% that had dementia. There were more female than male in the study, which is unsurprising for an Alzheimer's study. And the brain regions so far right now are middle temporal gyrus and dorsolateral prefrontal cortex. All right, so now actually using the Allen Brain Cell Atlas, what does that look like? So here is a screenshot of what the interface looks like. Here we're looking at the whole mouse brain data. I have the MERFISH data here on my left. I have the single cell data here on the right. On the left-hand panel here, you will see I have the class caret checked here. So I'm looking at all these different classes. And next to each class, there is a name and a colored box. And this colors, they correspond to the actual colors within the plots as well. So the pink here is our ITET glutamatergic cells. That's the same as the pink here in our single cell data. And this is what it looks like when you filter by class, and you filter things by clicking on the little ink droplets here. And these are our 34 classes that we have, and this is the whole mouse brain taxonomy. And also we have a couple of different Excel sheets and other tools to help use these data sets better, because sometimes it can be a little confusing figuring out what some of these acronyms mean. So if you click down here to brain-map.org, help and community guide cell types, we actually have a really nice spreadsheet that is sort of the key to understanding what some of these acronyms mean. So I would recommend having that open if you're unfamiliar with some of the names of the classes or subclasses. And here's how you would actually access the Allen Brain Cell Atlas. You can go to this link here. I also have it linked in this QR code. Or if you just search Allen Brain Cell Atlas, it usually is the first thing that pops up as well. We can also color by subclass. So again, we have 338 subclasses that are nested within those 34 classes. You can do that by clicking on this here, on this ink plot here, and clicking on subclass. And then again, since these are nested within each class, you can keep clicking on these carets over here, and they will reveal the subclasses, supertypes, and clusters that are nested within each group. And these colors, again, are the same between our MERFISH data and our single cell data here. We can also color by neurotransmitters. So the neurotransmitter type was also profiled for all of these cells. So here I did that by clicking on this down caret here for neurotransmitter, clicking on this ink droplet here, and I can see that all my glutamatergic cells are in blue. NA here means non-applicable, so those are cells that do not have any neurotransmitter expression. So a lot of our glial cells, our GABAergic cells here are in red, dopaminergic are in yellow, and there's a few other ones down here that are in smaller amounts. And we can also view genes. So I mentioned that there was originally a 500 gene panel that was used for these experiments, and that 500 gene panel was selected based on the single cell data. Here's what that looks like here. So not everyone's favorite gene is in the 500 gene panel. I tested one of mine here. So it's in the single cell data, but it was not present in the 500 MERFISH gene panel. And we actually have a list here that tells you which genes are in here, so you don't necessarily have to go and search all of them. But we also recently imputed these genes. So now we actually have, if you switch data sets and you go to here, so this is how you change your layout and your view here, clicking on this symbol up here in the upper left. You can add different visualizations, search through your data sets, and this one right here called imputed genes with reconstructed coordinates. This includes 8,000 imputed genes that were calculated from that single cell data and then now displayed in the MERFISH data. So the original MERFISH experiments were not done on these 8,000 genes, but we were able to take their expression level and that information from the single cell data and then put it into the MERFISH visualization. So now you can see those different genes, even though the experiments weren't actually done with those genes. And so here's what that looks like now. Now I have my imputed gene data set here on the left. Now I can search my favorite gene, SST, and color it. And now I can fully see it in the MERFISH expression in my spatial transcriptomic data, and also here on the right in my single cell data. And so again, we have 8,000 genes that were imputed, and you can find those here. We have them listed in our help guide right here, brain-map.org, help and community, guide cell types. So there's a lot of good information on here that's supposed to supplement and assist you in using this tool. So I just have a few more slides showing how to use the Atlas before I hand it over to Chris. And again, keep track of which data set you're looking at. It's labeled up here at the top. And so I can see that this is the one with the imputed genes. So I have more genes to search through in this data set. And again, you color everything by clicking on the little ink droplet here. Okay, so we also have different anatomical and dissection regions. So the 10x data, the single cell data was taken from micro dissections from 300 different mice. And so the displays here, the options are a little bit different based on the MERFISH data because there are some areas that are quite difficult to do micro dissections on that are too small. So the actual anatomical areas are slightly different in these two different data sets. And the data set here on the left, the MERFISH data, was mapped to the Allen Institute Reference Atlas and the CCF, the Common Coordinate Framework. So if there's any acronym here or brain area that you're used to seeing in the Reference Atlas, you can also find information on cell types in that area here as well, since it was mapped to the Reference Atlas. And again, you color that by clicking on the ink droplet. Now we have green here. Our isocortex is in green. We have our hippocampus here in a darker green. Our thalamus is here in red. And you can look at areas that are nested within different anatomical areas. So here in the MERFISH data set, we have the thalamus, but then we have subregions within the thalamus listed down here as well. And you can isolate and filter by different areas. So right now I just filtered by thalamus. So I have thalamus here in my MERFISH data on the left, and I check that box here. And then I also have it here on the right in my single cell data. These ones are different colors. Again, because I mentioned not every anatomical area was possible to have a micro dissection associated with it. So we have different colors and they don't 100% match up for these two different data sets. But I both have thalamus checked off here so you can see what they look like both in the single cell data and in the MERFISH data. Okay, and then when you have your different filter set, you can also search by different genes. So I typed in calbindin. Now I'm looking at calbindin just in the thalamus. And I do that by typing in a gene, clicking on that ink droplet. And you can also have two different genes side by side. So here I still have the thalamus checked off. I have calbindin or calb1 on the left, and then I typed in calb2 or calretinin on the right. So you can duplicate your screens here, and you do that by clicking on this little plus button here. And because currently there's no way to look at co-expression between genes in the ABC Atlas, but you can have two different genes side by side, or you can toggle between the two in the same view. And what are some use cases? You can see, again, I mentioned you can't directly look at co-expression, but you can have two panels side by side or toggle between them. So that's one way you can look at, are two different genes co-expressed in the same cell type. If you want to look where a certain gene is expressed in the brain, is that gene expressed in a particular cell type in the taxonomy in the brain? If you're studying a certain cell type and you want to know where it's expressed in the brain, and also the spatial relationships between different cell types, since we have that spatial transcriptomic data as well. And just a few caveats, what you're seeing in the MERFISH data is based on segmentation of the individual somas of each cell. And so we're only looking at the transcripts within each soma. So if you're interested in looking at synaptic contacts or any cell-cell interactions, that's not possible with these data and the ABC Atlas. And also, since we're just looking at the cell segmentation, and the MERFISH data is just showing those individual little dots. Those dots actually represent the centroids of the cells. And when you zoom in and out, you will see that they actually change shape and size. So all of our cells are not perfectly round, and they're not all changing shape. It's just because each dot is actually representing the centroid. We do have the raw MERFISH data that was used to create, the MERFISH the plots here in the ABC Atlas. So if you're interested in the raw MERFISH data, we do have that accessible on our website, and then you can see more of that. But yes, so these are some things that are currently not possible to answer with the data set and how it's set up in the ABC Atlas. And there's some other things that are currently not possible to answer in the Atlas as well, such as certain types of calculations or quantifications, which is why we have Chris here, who can show us how to access these data sets programmatically and do some more difficult or some more a bit more advanced computations with these data sets. And so now I'm going to hand it off to Chris.
Thanks, Rachel. Just get my screen going. Okay, Yeah, so as Rachel said, I'm going to walk folks through sort of at high level the tools that are available for programmatic access to these data. This is not going to be a Python tutorial, unfortunately, if you're not familiar. All of this stuff is written in Python and uses Jupyter notebooks to basically recreate a lot of the plots you saw Rachel present from the ABC Atlas visualization tool. But with the actual data in hand, you'll have a lot more access to, analysis you want to do. So let me start this up. Okay. So this all exists as a GitHub repository in our Allen Institute account, ABC_Atlas_access. And it basically provides three things for the community. One is a Python object that allows you to download and keep track of versions of the data that you're downloading. So we've released we've had several different releases as Rachel's gone through, like incrementally adding data or improving data that has been released. So this will track whatever consistent version you're on, and also keep track of which files you've already downloaded. So if you want to work offline, once you have the data in hand, you'll be able to do so. It also provides a set of Jupyter Notebook tutorials to basically try to orient you on how to interact with the data, going from the metadata to the H5AD files storing gene expressions themselves, and also, as I said, showing how to recreate some of the plots you saw in the ABC Atlas. And then finally, it has a render of all of this information as a static website that I will show you right now. So here's the GitHub repo. If you also want to get to the website that's linked down here, basically a description of what tool is for and what this tool is not for. But here's the website. It's alleninstitute.github.io/abcatlasaccess. So this has a description of all the data, of what the tool is and all the data that is currently available. So you notice there's only 4 data sets listed here, as opposed to the 5 that Rachel went through. We currently don't host the CAD data through this tool. But if we go through, we can see descriptions of basically all of the actual individual data sets that make up in this case, this is the whole mouse brain data sets, usually abbreviated internally by us as WMB. So as you see that around, think whole mouse brain. So each one contains a description of what's actually in the data set. This is the page representing the full metadata for this data set. And conveniently enough, it also has a link to the actual data in S3. So if you were to click through here and download the files, you can do so individually. But we do recommend you use the Python tool, the Python library to keep track of everything and give yourself a consistent version. So as I said before, there are tutorial notebooks that come with these. So if I just click on a quick one in the mouse brain, if I scroll down, you can see that come on. There we go. This one isn't showing that, but here's basically the taxonomy as a cute little ring graph. Let me grab one that actually shows the ABC Atlas plots. There we go. So you might have recognized this from the slides that Rachel just showed, and this is all data you can download and visualize yourself instead of relying just on the ABC Atlas. Cool. Back to my presentation. All right. So helpful links to get yourself started with this data set. So as I said before, it requires Python or just some knowledge of using Python to interact with not only downloading the library and installing it, but also interacting with the data itself. I typically use Miniconda, which is a sub-release of Anaconda. There's other options out there to keep track of environments and keep track of your Python. So I'll leave it to you if you have your favorite or if a colleague has their favorite. What I'll be going through today are Jupyter notebooks. So you'll need Jupyter for that. This is just a nice tool for interacting with with the data. If you ever use like MATLAB or Mathematica, it's very similar. And finally, one of the main things, all the data is in CSV files or is basically in CSV files, and we load those through pandas and manipulate the data with pandas. So some familiarity with pandas will eventually be needed for you to actually to run through the notebooks and do analyses yourself. But there's plenty of tutorials online to help you get oriented. Started with Python, started with pandas. So the hope is you can get up to speed fairly quickly using the tutorials that we have. And finally, there's just links to the website, GitHub repository, and the notebooks themselves. Great. So what am I going to be walking through today? So first thing I'm going to show is just the Getting Started notebook. This is basically a notebook that hopefully can orient you on the tool and also explain how the data is structured and how to download and re-download data. Next, we're going to go look at some of the whole mouse brain data, the WMB data, just the 10x though. There are tutorials that show the Merfish off, but we're just going to go through the 10x today. And then after that, we're going to select genes that are expressed in subtypes, or I guess those would be supertypes, or a level below the subclass that is L5-ET-CTX glutamate. So this is a specific subclass in the taxonomy, and we're going to basically explore it a little bit. We're going to plot those genes we selected in a heat map as well as in the UMAP. And then finally, since Rachel was showing off the ABC Atlas visualization tool, I will go through a quick example of how, if you have some interesting cells that you're looking at or an interesting region in that that visualization, how would you translate between that and the ABC Atlas Access data on your disk? So with that, let's get started with the Getting Started notebook. So basically the tutorials assume that you're not too familiar with Python for the most part, and do set you up to be able to install the package at least. So once you have Jupyter installed, if you fire up the Getting Started notebook, this command here will install the library in a configuration that's necessary to run these notebooks. So they'll install pandas, NumPy, SciPy, all of everybody's favorite things to access the data. And then once that's installed, there's a line to restart the kernel so that you'll be ready to go. And Rachel assures me, as a non-Python expert, that she was able to get through these notebooks. So thanks for testing them out, Rachel. All right. So once we have the package installed, we can load the package, which is just ABC project cache. And then once we have that loaded, we need to specify basically location on disk. So this can be anywhere. We've designed the tool with in mind that you might be able to run this on laptop scale, but be forewarned that unless you have a large hard drive, you will run out of space on that disk very quickly if you start downloading a lot of the gene expression data. So for instance, for this tutorial, I've downloaded all of the WMB gene expression data, and just one of those directories is 100 gigabytes. So my laptop's hard drive has already screamed at me a few times since setting up this tutorial for a workshop previously and this one here. So be forewarned with that. So here I've just specified a relative directory to where I'm running this notebook to download and run the data, or download and store the data. So we see that we are currently on release 2025-01-31. So this is the release that includes the aging mouse datasets. And if you run a quick command, you can list all the versions that are available. Now, these do build on each other. I probably only recommend going back to a previous version or keeping with a previous version if you've already been working with that one since the beginning. So just have a stable version if you're trying to say, write a paper on this specific release of the ABC Atlas, using the specific release of data from the ABC Atlas. If we do want to go back in time, though, we can just specify the name of the manifest and load that. And we can see we can go back in time here's an older version, or we can, but for this tutorial, we'll switch to the latest. For the cache, you can list all of the dataset directories that are available. Now, if you remember from our whole mouse brain set, there are all these different directories here. These are basically matched in these in this list. So for the whole mouse brain 10x data, you can see that there are multiple directories here. So there's 10x multi v3, v2, and then as well as metadata on the taxonomy, the cells themselves, and so on. Well, actually, that's the human mouse brain. This is the mouse brain, human mouse brain. Sorry, it's early. Okay, so once we have, say, a directory, we can list the data files that are in those. So this is a list of all of the available H5AD files that we could download from the whole mouse brain. The whole mouse brain data is split up by assay and dissection region. So you can see different cuts of the isocortex and the thalamus and midbrain. So these are what we refer to as data files. So those are all H5AD files. And then metadata files are all our CSV files. So for the WMB taxonomy, you can see there's definitions for the clusters, how they're annotated, and various mappings between the cluster and the color that we've given it. If you want to know how much data you're going to be downloading, if you were to grab full data sets, so for just the 10xv2 data, that's 100 gigabytes worth of H5AD file. However, the metadata metadata directories tend to have much fewer files. So unless you need to interact with the gene expression data, save that until you absolutely need to. The releases basically have metadata to sort of guide you around and interact with the cell type taxonomies themselves. And then once you're up to speed on that, you can start downloading H5AD files and playing with gene expressions. And then from there, it's a simple command to download the data. You can either download all of the data at once in a directory. So I'm not going to run this again because this data is already downloaded. But you can see here, there's a progress bar. The code will even check the hash to make sure that you've downloaded the data properly, and it's the stable version that we expected. But you can see if I run this again, it doesn't have to download anything. It already knows the files are on disk and waiting for me to interact with them. So you can get all the metadata as well. So this is just getting all the taxonomy data we listed. And yeah, this is just proving that it doesn't re-download. You can also download individual files. So if you have a large data set that you just want an individual file for, you can specify it by the directory and name. But this is all tutorialized online, so I'm not going to go complete detail. Yeah, the one thing to note here is that there are two different versions of each H5AD file. One is log2 normalized and one is the raw data, and that needs to be specified when you call the name of the file. Else you get an error that it is at least helpful telling you that you need to specify at least raw or log2. There are advanced options for this in terms of forcing re-downloads and skipping hash checks if you want to bulk download things, but we'll get for that for now. Okay. So let's go over a quick demo of things you can do with this data set. So we're interacting with the whole mouse brain. And grab some gene expressions for L5ET CTX GLUT. So here's again, we can see we're instantiating our ABC Atlas cache, and some stuff will need like pandas and utility functions and plotting. So we'll just specify where our cache is. We're downloading it from S3 and just using the current manifest. So to interact with the gene expression matrices, we have a convenience function that will download the files that are needed and interact with the H5AD files one gene at a time. If you've ever looked at gene expression data, you know that they can blow up very drastically in your memory. Our H5AD files are no different. They are stored as compressed matrices. So they will explode in memory if you try to load individual genes out of them. So it's best to iterate through them, but we do have a tool that will uh, basically abstract all that process away for you. But to use that and extract these genes that we're going to get for L5ET, we first need to load the cell metadata. So this file specifies for each given cell what file it is in, various other metadata like libraries, unique identifiers, and for this specific data frame, it also contains the color information. So this is the neurotransmitter color, the class color, and so on. And these are the same colors you'll see in the ABC atlas. Here they're just presented as hex codes. But yeah, I'll specify what subtype, cluster, class it is, what donor it came from, the region of interest the cell originated in, and so on. So this is basically your one-stop shop for information on a given cell. We can also load the gene panel. So this is the full set of genes that are in the 10x data. I've just shown the first 5 here with the ensemble IDs, the gene symbols, expanded name, and if we've been able to map them to the NCBI, an equivalent NCBI gene that's also there. Okay. So we have our cell metadata and our genes ready to go. So let's specify some genes we want to extract. So here are marker genes for the supertypes that are just below the subclass that we're interested in. So these are our marker genes that differentiate between the different subclasses, supertypes, sorry, within the subclass. So we have a convenience function here, get gene data, that takes in our ABC Atlas cache, our cell metadata, our genes, the genes we want to select, the data type we want to load, whether it's log2 or normalized or the raw counts, and then a chunk size. So this is basically specifying load this number of rows per time that I iterate through the data. If you find yourself running out of memory, say you have a small laptop, or if you're working in the cloud and don't have haven't purchased the most robust instance, you can reduce this chunk size and it should eventually work. Okay, I'm not going to run this now, but here's the call that I have set up for this notebook. So the ABC cache, the cell matrix I loaded, the gene matrix, the names of the genes I want to load, and a chunk size here, what I just specified 8,000 just by the default. So this takes about 10 minutes to process. It goes through each and every file and loads the given rows, selects the genes out that you want, and then returns them to you. And this is all necessary because these are sparse matrices, stored as sparse matrices. So if you tried to load one of these files on its own and slice on gene, your kernel in Python would crash because it would run out of memory, unless you're on on a very large cluster. You can see this takes about 10 minutes to run. Probably take about like 2 hours if you're trying to download all the data at once, because as I said, there's a significant amount of data and it depends on your download speed. Once that's done processing, you can see we have gene data. So for each cell from that matrix with its unique identifier, we now have the expression levels of our 5 selected genes. Okay. But fortunately for me, I have pre-baked basically a gene data expression. So we have workshop demo genes at CSV that's just available that I've already run ahead of time. So I'm going to merge that with the cell. So now we have a pandas data frame that's cell by gene. Cool. All right. The other thing that is in that cell matrix is this position of the cell in the UMAP. So we can just declare a convenience function here. Hopefully I've documented everything available in this function. This is just basically an example of I want to plot my given cells. So that's input data frame. Let me get that input data frame. And and then extract a given subclass or supertype or some sort of cell type taxonomy and plot it on the UMAP. So if I scroll down and I execute this function with my cells that I'm running, I'm going to plot them by, or color them by the subclass cell type. And you can see here, it's very familiar to what you just saw Rachel present. And then with my convenience function I've declared, I'm going to select the given cell type that we have, which is this one. And lo and behold, we're coloring all this other stuff gray, and then here's the actual cell type of interest. And now let's go look through and look for each one of our genes in the names we selected. So starting with CRYM, we're going to plot the same UMAP, but this time color by the gene expression. So again, this is very similar to what Rachel presented, where you have one selection in the ABC Atlas and you're coloring by a gene. She did it as a, she showed it as a region of interest selection. This is a specific cell type. So you can see here for the first gene expression, the next one, and so on down the line. And you can see how these genes are fairly isolated in a given portion of the UMAP, which makes sense as they are, markers for a given supertype that is below the subclass level in this taxonomy. All right, we can define other convenience functions to basically average for a given metadata selection. Say we want to see, the gene expression level for a given region of interest or other cell type selections. So we're going to declare functions to aggregate that metadata, just basically using pandas group files, and then plot those functions using matplotlib as we did above. So we're going to just look at cells in L5ETCTX by selecting the specific subtasks from our main data. This is just Python slicing and masking, if you're familiar. Once we run our aggregation, we can plot, which is just averaging for each given selection we have the data, the expression. We can plot the heat map, and you can see here that, yep, there are definitely these genes determine what the supertype is. So if you look at realm, there is a high expression in this specific GLUT4, which I'm going to very strongly suggest that this is actually the marker for GLUT4. So this is what one of that averaged set of data looks like. You have the genes by the supertype, and this is their average expression value in log2 across those. But we can specify more and go deeper. So this is those same genes, but for the clusters underneath our subclass. And I believe I've coded it up to allow for basically any sort of selection up here. So if you were to have the full data frame, you could do region of interest as well. All right, then finally, really quick, let's say you've been playing around with the ABC Atlas itself, and you'd like to look at what those cells, what the actual data for those cells are, not just in the visualization, but in the gene expression matrix or in the metadata. So in this example, we've taken cells from that same subclass, this time under a supertype under it. We've selected 84 of them. Note that when you do select stuff in the ABC Atlas, you can only select them 100 at a time. So this is probably more useful for MERFish data, which has specific regionality to it. But, here's the actual atlas. If I select cells, you'll see a little pop-up over here. And then there's a download button that you have available. And this is basically the same region-ish. So once we have those cells selected, we can download that as a file. So that's this button over here. I believe it, yeah, download first 100 cells. And that'll be just downloaded as a CSV onto your system. If we read that using pandas and look at this data, we can see there's a unique identifier that's not quite the same as our cell types, our cell metadata identifier, but it has most of the same information as our metadata table above. Uh, but if we do load our cell metadata, we can see that those IDs are now available in these metadata files. So uh, using pandas, we can join and then mask on those cells, and we can see that from this cell metadata table, which has about 4 million cells in it, we've now selected just 86. So these are a subset of the cells that we've actually selected now with the joined columns from the ABC Atlas visualization. And then finally, if we select those out, we can plot them again using our UMAP, and we can see a very extremely tiny purple splotch on the UMAP that says, yes, these are the same cells that we've selected back here. If I were to zoom out on this, but this is a static image. So basically somewhere around this region. So yeah, all of this code and notebooks are available online. We have demo links to this specific demo that I've shown on the GitHub and a Google Drive that has the example genes on it, as well as copies of all the notebooks and the other things that are downloaded for this. Even if within that Google Drive, there is an example link to an example in a text file. There's a link to an example implementation of running this in Google Colab, mounting the public S3 bucket using a software called S3FS Fuse. So if you are running on the cloud, there is an option where you don't have to download hundreds of gigabytes of data yourself and mount it. You can mount the data itself on S3. So with that, I'll stop and head back to Rachel.
All right, thank you, Chris. And as he mentioned, I'm not someone with a strong programmatic background, so I tested out these Jupyter notebooks before we release them, and if I can get through them, I have faith that others can get through them as well. They're pretty easy to follow, and I'm glad that Chris has made them. Okay, so now I'm just going to share my screen once more, and we have a couple minutes for questions here. We do have about 5 more minutes till the end of the webinar. So if there are any questions, we can take them now. And we also do have a feedback survey as well. So I'm going to launch that. And so that you should have a feedback survey on your screen. And this is just really helpful for us figuring out how useful these webinars are, what things could be added, what things could be improved. So if you could take a moment or two, and this is all anonymous, so you can put whatever you'd like in the survey. We have no way of tracking who's answering what. So I'll just leave this up here for a moment.
And if you do find yourself with questions regarding the data available through the tools, as Rachel pointed out, post on the community forum about it, ask questions. Those questions will eventually get to me and help you walk through the notebooks if needed.
Yes, yeah. If you get stuck at any point on any of our tools, but especially with these ones, yeah, the community forum is heavily monitored by many groups of scientists at the Allen Institute. So someone will likely try to answer your question and try to help you along if you have any barriers or points that are preventing you from doing your work.
And if you find a bug or issue, please post it onto the GitHub as an issue.
If anything's not working for some reason, that's also a very useful feature of the community forum. People are quick to let us know when something's not working. So that's very helpful for us. And I mentioned those data sets in the beginning, the whole mouse brain, whole human brain, Alzheimer's, and the aging mouse brain. And there are other current ongoing projects at the Institute that once they're published, those will also be added to the ABC Atlas as well, and then also have accompanying Jupyter notebooks. So we are not limited to those data sets. There will be many more data sets and other topics that will be added to the notebooks and the actual atlas itself throughout the year. So keep an eye out for that.
Mhm. Yeah, and additionally, the website I showed. So the notebook that I ran through for this webinar is basically a concatenation of several different notebooks across. So if you are lost, or there were too many steps in that one, each section that I presented has its own walkthrough, effectively as a notebook on the website.
And they're pretty similar between data sets, right? Like I noticed that when I was following them, you're making certain plots or doing certain things in the whole mouse brain data set, but if you're interested in the human data, those notebooks are pretty much set up in the very same fashion. So if you know how to get through one and you can navigate that, it's the same pretty much in all the other ones.
Yep. We try to keep the metadata and H5D files similarly structured. So what applies to one will mostly, with some caveats, apply to the other one. It depends on the data we're actually handed.
And this is helpful for doing some of those computations, calculating gene expression, maybe doing heat maps and, answering those questions that are currently not possible to answer using the GUI of the ABC Atlas itself.
Yeah, as Rachel mentioned, co-expression. We presented a lot of this stuff as a workshop last week, and immediately someone was already trying to do a co-expression with this data. So this is something that allows you to go beyond the atlas itself.
And if there's any, feedback that you have on features you wish there were, or other things that's also helpful to let us know in the community forum, because our data and technology team is constantly working on improving this tool and adding new features to it, getting feedback from the scientific community. So if there's something that's you think would be useful that is currently missing, that is also very helpful information. So feel free to mention that somewhere on the community forum as well. Okay, and I'm going to end the poll now. Thank you all for answering that. And I will just jump back to just remind you all that this is part of a webinar series. So we have had some previous webinars in the past. Again, we have that aging mouse brain data set we'll be going over on April 23rd in about a month. You can register for those at alleninstitute.org/events or use this QR code. And we will have another webinar coming in May and also some virtual office hours. So we have not scheduled them yet, but we will be announcing them soon, where we'll have some of our scientists here on hand just to answer questions you have about using certain tools or certain data sets. And again, here's our link to our community forum, community.brain-map.org. And we just want to thank our funding for this. This webinar series is sponsored through a grant through the National Institutes of Neurological Disorders and Stroke. And yeah, thank you everyone for joining us for today's webinar.